The School of Biomedical Sciences is one of the largest teaching and research schools in the Faculty of Medicine and Health here at University of New South Wales. We develop and deliver teaching to undergraduate medical, biomedical and science students. And our research seeks to understand human health and disease at the fundamental mechanistic level. One of our key research pillars is biophysics, which is represented by a constellation of researchers who are using biophysical techniques and approaches to understand the mechanisms that drive human health. One of the areas that we're particularly interested in right now is what's going to happen to our bodies when they're exposed to the unfamiliar condition of weightlessness in space. There, the ability of cells to sense forces is disrupted because all of those force sensing mechanisms have evolved here over 4 billion years under the conditions of Earth's normal gravity. We think one of the really key things that we need to understand about cellular force sensing is actually exactly where these molecular force sensors are being activated. So we study a particular type of force sensor, the mechanically activated ion channels. These convert mechanical information into electrical signals, and the patterning and timing of those signals at the surface of the cell is really key for the effect they have on cellular function.
We try and look at single molecule behavior because these behaviors are important to not just maintaining healthy cells, but also understanding what goes wrong in disease. Single molecule imaging enables us to visualize these molecules in real time as they perform their functions in living cells. And this allows us to then interpret what happens when cells signal and adapt and when these go wrong during disease. Our view is that for us to understand what goes wrong in disease, it's important for us to understand what is healthy actually looks like. And while there's tremendous value in translational research, I think what is really important is to have simultaneous fundamental research that can give us new targets for therapy as well as open avenues for future diagnostics and therapy.
Being able to visualize how individual complexes come together, seeing the formation, seeing the heterogeneity within these signaling complexes gives us an entirely different perspective on the biology of a cell. Developing new microscopy techniques has been critical for that, especially super resolution microscopy, because that allows us to visualize these processes as they happen on the scale of the individual molecules. The real strength of super resolution imaging is being able to translate complex immune cell behavior into specific parameters, how the complexes form and why that translates into signaling processes. That allows us to pin down what are the critical parameters for activating a T cell or how a T cell makes those decisions and makes appropriate responses. And that gives us the targets that we need to design new therapeutics.
In order to be able to actually see where these molecules are activated in the surface of the cell, we need advanced imaging tools that allow us to visualize individual molecules and their behavior. One of the amazing things about working here at UNSW is we have access to cutting-edge instrumentation that allows us to resolve these individual molecules and dynamics in complex cellular structures.
In order to be able to design and engineer living systems, we need to understand the physics of living systems first. The way to think about the role of theory is to look at how it works in a theory-centric subject like physics. And there, the main way it works is by taking concepts and ideas from one system and trying to extract them to make predictions for another system. And so if we can draw out core principles and core ideas in living systems, then it helps us generate hypotheses for our research in other areas. I see interdisciplinarity in a very straightforward way. You take two people who both have expertise in different areas and you add them together and you get twice as much expertise.
Our approach incorporates expertise from different disciplines to be able to record molecular movies of molecular machines. So we have biochemists who make proteins and rebuild the machines from bottom up. We have chemists who devise new fluorescent probes, and we have physicists and engineers who build microscopes with higher resolution and image analysis algorithms for us to be able to extract the information from these molecular movies. We can put all of this together to generate mechanistic insight that we can then go to our cell biology and virology colleagues to test these mechanisms in the complexity of the cell.
As part of my role as a teaching and research academic, I'm not only running a research program, but I have the pleasure of teaching undergraduate students. For me, that opportunity to link existing knowledge with new findings and communicate to those undergraduate students how cutting-edge research can advance what we know about human health is a really inspiring part of my job.
Being at the forefront of researching biophysics also gives us a good perspective on teaching it to new students. And I think this is critical, and we're designing our new biophysics major with the view that biology now is multi-scale and quantitative.
We're really excited to launch the new biophysics major. This biophysics major will enable students to get a comprehensive training in data-driven and quantitative field that biophysics is. We will first ensure students get foundational training in the principles of biology, physics, and chemistry, which underlies all of biophysical research. We will then ensure that they have a problem-based approach to tackling contemporary biophysical problems using biophysical techniques. Viewing through a biophysical lens enables us to understand how human health is governed by physical principles such as forces, transport, signaling, and energy, and understand how the same processes can go rogue in disease. This also opens us avenues to understanding how we can develop new diagnostics or therapies for disease in the future.